Look closely at these glowing segments of DNA turning on and off. We often treat our genetic makeup as a fixed biological destiny, a hand of cards we're forced to play. But the science of epigenetics reveals that our genes are equipped with biological switches. Environmental factors like pesticides and food additives operate these switches. Exposure to these chemicals can silence the genes that protect our liver or activate the ones that cause disease. To see how these biological systems can be sabotaged, look at a disorder called hemochromatosis. It serves as a baseline example of a body out of balance, forced to absorb massive toxic amounts of iron. This diagram shows normal cellular absorption on the left, but on the right, a flood of unbound iron molecules acts as a powerful cancer fertilizer. This free iron triggers intense free radical damage and stimulates chaotic, rapid cell reproduction inside liver tissue. Your biology is a switchboard, not a stone tablet. The unseen, daily exposures in your environment dictate whether your liver's defenses hold strong or fall. Some of the most destructive environmental saboteurs are completely invisible, like the highly toxic fluoro-aluminum complex. This compound forms when standard municipal water, treated with fluoride, combines with the aluminum found in everyday items like soda cans, baking powder, and common cookware. Even standard black tea carries this risk. As this diagram shows, the root system of a black tea plant naturally acts as a sponge, absorbing and storing high concentrations of fluoride and aluminum from the soil. These invisible exposures extend into the produce aisle. Rinsing your salad greens with commercial veggie wash offers zero protection against many modern agricultural chemicals. Systemic pesticides like glyphosate are absorbed into the very structure of the plant. Because the chemical is inside the tissue, it is impossible to wash away. This creates a double threat with genetically modified soybeans. Soy naturally contains the estrogenic compound genistein. When combined with heavy glyphosate, the plant becomes a contaminated hormone disruptor, bypassing EPA safety limits. These agricultural practices are engineered to bypass surface-level cleaning, turning common foods into hidden endocrine disruptors. Beyond synthetic chemicals, certain staples often found in traditional health diets can secretly burden the liver. Let's look at whole grains. Whole grains deliver inflammatory lectins and excitotoxic glutamate, compounds that damage the intestinal lining and add to the liver's toxic load. This split screen illustrates a similar trap with fruit juice. On the left, whole fruit digestion is paced by fiber. On the right, fruit juice delivers a concentrated liquid strike of fructose directly to the liver without any fiber to slow it down. Dairy, whole grains, and fruit juices often act as dietary Trojan horses, driving fat accumulation in the liver and stimulating the growth of disease. When this damage causes liver cancer, the resulting tumors use a specific architecture resisting conventional treatment. Dr. Russell Blaylock uses the metaphor of a child's bubble blower to explain this. The wand is the source and the bubbles are the result. In actual biology, the bubbles are daughter cells and the wand is the cancer stem cell. Chemotherapy pops the daughter cells but fails to destroy the stem cell. This surviving cell alters its metabolism, consuming massive amounts of glutamine, converting it to glutamate to accelerate growth and invade tissue. Because conventional treatments target the expendable daughter cells rather than the stem cell source, the cancer inevitably returns with multi-drug resistance. To counter this, we look to nutritional protocols designed to flip epigenetic switches and dismantle the tumor's metabolic architecture. The plant extract berberine offers a specific mechanism to combat this metabolic hijacking. Watch the cellular transporter mechanism in this animation. Berberine physically blocks the transporter that allows glutamine to enter the cell, essentially starving the liver tumor of its primary fuel. While chemotherapy focuses on daughter cells, research into natural compounds like nanocurcumin, nanoquercetin, and pterostilbene shows they specifically target these elusive cancer stem cells while protecting healthy tissue. By cutting off the glutamine supply lines and destroying the stem cell bubble blowers, these natural compounds address the metabolic source of the disease. Even in extreme cases, like end-stage liver transplants, the risk of organ rejection and infection remains a massive clinical hurdle. A clinical study demonstrated that a simple intervention could change these outcomes. Transplant patients given an emulsion of omega-3 oils, specifically DHA, showed dramatically improved survival rates. DHA inhibits the inflammatory cytokines responsible for organ rejection, leading to shorter hospital stays and a lower death rate compared to those on a standard hospital diet. Swapping black tea for white, removing dietary Trojan horses, and utilizing targeted nutrients puts you in control. You are not a victim of your genetics. You have the power to operate your epigenetic switches and restore your liver from the ground up.